Hey guys, welcome to my YouTube channel. If you are new here, you are very much welcomed. Kindly go and subscribe to my channel. Like and comment to my videos, share with your friends and family for more content. So in this video I'll be sharing with you the process of protein synthesis, including the transcription and the translation process. I made this video to be very short and easier for you guys to understand, so I hope this video will be useful to you. If you have any questions regarding this topic, kindly go and type on the comment section and then I'll get back to you. Bye, enjoy the video. Good day everyone. In today's topic we're going to focus on protein synthesis. The key word here is the word synthesis. To synthesize means to produce or to manufacture something. So in this case it is clear that we are manufacturing or we are producing the protein. Now the protein synthesis, it refers to a process by which proteins are made or produced in each cell of an organism. Now, let's take a look at the concepts that are involved in the protein synthesis. We have the four major concepts here that we need to understand them into detail. The first one is the mRNA, the tRNA, the transcription as well as the translation. Let's take a look at the first concept called the mRNA, the messenger RNA. So this is a single-stranded RNA that is involved in protein synthesis, and it contains what we call the codons. Let's take a look at the structure of the mRNA here. This is the structure of the mRNA. You can see it is a wave-like structure carrying the information copied from the DNA, and these triplet bases, the AUG, the ACG, these are what we call the codons. Our second concept here is called the tRNA. So the T here stands for the transfer RNA. This is a single-stranded RNA that helps to decode mRNA sequence into a protein, and it contains a triplet of bases called the anticodons. So remember, on the mRNA we have the codon, so here we have the anticodons. Now its function is to bring the required amino acid into the ribosome. Let's take a look at the structure of the tRNA. It's represented here on the diagram in a yellow structure here. So this is a tRNA. At the bottom you can see we have the triplets of bases called the anticodon, and at the top, at the receptor structure of this tRNA, we have the circles containing what? Containing the amino acid that are required by the cells in the ribosome. So the example of the amino acid here that are found is the aspartic acid, the lysine, as well as the tryptophan. The next concept is called the transcription. So now what is a transcription? A transcription is a process by which information in DNA is copied on to a molecule of mRNA, and this process of transcription takes place in the nucleus of a cell. The next concept is called the translation. So now what is a translation? A translation is a process by which a protein is produced using RNA molecules, the mRNA, the tRNA, as well as the rRNA. And this process, it occurs on the ribosome of the cell inside the cytoplasm. Now let's take a look at the process of transcription, a process in which DNA replication takes place and a process in which information is being copied from the DNA molecule. This whole transcription process occurs in the nucleus of a cell. So in a transcription process, we have the DNA molecule, which is our double-helix structure. The first step, double-helix structure unwind. Weak hydrogen bond between the complementary bases break. Now we call it a weak hydrogen bond because it can break any time and separate these two strands. Now the two strands separate. One strand is used as a template, and then free nucleotide bond with the complementary bases of the two strands. Now our new DNA structure is formed. This is the mRNA structure, a new DNA structure carrying the information from this DNA. After this process of transcription, this mRNA, it moves from the nucleus of a cell through the nucleopores into the cytoplasm, where it will attach into the ribosome and start the process of translation. Let's take a look at the process of translation. This is the last process to synthesize the protein. Now, the process of translation, it occurs in the ribosome of a cell inside the cytoplasm. What is happening during a translation is we have the mRNA, remember, that has the copied information from the DNA replication. The mRNA, it comes into the cytoplasm where it attaches into the ribosome. There comes the structure of our mRNA attaching into the ribosome. Next, we have the tRNA, the transfer RNA. Remember, the purpose of the tRNA is to bring the specific and required amino acid into the ribosome. Now there comes the structure of the tRNA. The tRNA comes and attaches also into the ribosome to bring those specific and required amino acid. Remember, the tRNA, it carries the anticodon, while the mRNA, it carries the codon. When the tRNA attaches to the ribosome, its anticodons matches the codons of the messenger RNA, and then it brings the required amino acid. Now at the top of the tRNA structure, there are amino acids attaching in the receptor structure of the tRNA. Those amino acids together, they are joined by a peptide bond. So a peptide bond joins those amino acids together to form the protein. So this is how the proteins are formed. Now, to clear the misconception in this process of translation, we are not synthesizing the amino acid here. We are using the amino acid and joining them with a peptide bond to form the protein. Thank you.